Throughout this lesson, we have focused on the role of enzymes, enzyme structure, and in particular, HIV's reverse transcriptase and its role in HIV replication. The end goal is how do we stop HIV replication, which starts the discussion on how can we control enzyme function and reaction rates. The purpose of this lesson is to give you a brief overview of these different environmental factors that we can place upon our enzyme to control their function and reaction rate. In certain cellular environments, enzyme activity is partly controlled by environmental factors like pH, temperature, salinity or salt concentration, and in some cases, what we refer to as activators, which can be our cofactors or coenzymes. We can also have the discussion on enzyme concentration, substrate concentration, and inhibitors. So starting with enzyme concentration, if we look at our graph here, we have reaction rate on our y-axis and enzyme concentration down here on our x-axis. And the idea is if you increase your enzyme concentration, you're also going to increase your reaction rate. It's directly proportional. And the reason that is because you have more enzymes that can collide more frequently with substrates. But eventually that reaction rate will level off because your substrate becomes a limiting factor. And what that means is you have more enzyme than what you do substrate. Substrate concentration is very similar. If we increase our substrate concentration, we can also increase our reaction rate. So the more substrate you have, the more collisions that you're going to have with enzymes, which again, just like in the last slide, will cause your enzymes to become a limiting factor. Therefore, your reaction rate will level off. That moves us to temperature. And again, we're looking at the same type of graph. However, the difference with this, because temperature happens to be an environmental factor, which we know that we can increase and decrease temperature, which means we're going to get more of what we call a bell-shaped curve, which means up here at this point, if we go down, this point right here on your graph is going to offer what we call an optimal temperature. And in the human body, the optimal temperature is 37 degrees Celsius, which would be normal body temperature of 98.6 degrees Fahrenheit. This is where you're going to have within the human body with enzymes the greatest number of molecular collisions. So the idea is you can go one or two ways from your optimal temperature. You can either increase it or you can decrease it. Either way, whether you increase or decrease your temperature, you're going to notice that on this graph, your reaction rate is going to decrease. So basically, an increase in temperature beyond your optimum temperature, there will be an increase in energy which disrupts the bonds or your intermolecular forces within your enzyme substrate complex. And what that's going to do is result in denaturing your protein, simply meaning your enzyme is going to lose its 3D shape. And if it loses its shape, it's going to lose its function. On the other hand, you can decrease the temperature, which is going to cause your molecules to actually move slower, decreasing that collision between enzyme and substrate, which will result in the same thing of decreasing your reaction rate. That moves us on to pH. pH is another environmental factor. If we think here of a pH scale, we have 0 to 14. So here would be about 7. And again, just like temperature, we get these nice bell curves. And this we do. And what happens with pH is, again, we have an optimal, well, that just kind of bothers me. We have an optimal temperature. So up here at this point, we're going to have an optimal pH. Just like here, we're going to have an optimal pH and an optimal pH. So in humans, our enzyme's optimal pH is anywhere between 6 and 8. Protease, which is one of your enzymes with HIV, has an optimal pH of 4 to 6. And again, if you increase or decrease your pH, you're going to disrupt bonds or the attraction between your charged amino acids, and that's going to disrupt your 3D shape of the enzyme and will result in denaturing your protein, so therefore making that protein non-functional. We also get into salinity. Salinity is a way of saying salt concentration. And again, we can have a salt concentration at 0 up to some number here. In humans, we like our salinity or the salinity of our blood to be about 0.9%. And again, if you notice, we get that nice bell shape where we can increase and then it begins to decrease. So if your salt concentration is close to 0, the charged amino acid sides of the enzyme molecules will attract to each other. This will denature your enzyme and form an inactive precipitate. On the other hand, if your salt concentration is way down here, or it's too high, the normal interaction of your charged groups will be blocked, new interactions will occur, and again, your enzyme is going to precipitate. So an intermediate salt concentration, like what we have here at 0.9%, is going to be optimal for most or many of your enzymes. So another factor that can affect enzyme function are what we call activators. Activators are your non-protein helpers that basically aid in that catalytic activity by increasing your reaction rate. So the faster these reactions can occur, the more the enzyme can be used. Because if we think back to our last lecture, we know that our enzymes are not consumed in a reaction. So your activator is just simply going to help your active site become faster. And we have two types of activators. We have what's called cofactors. These are your small inorganic compounds and ions that are bound within the enzyme molecule. A popular one that you guys might be aware of is with hemoglobin. We have the iron that is bound within there. We also have coenzymes. Those are your organic molecules that can bind temporarily or permanently to an enzyme near its active site. And these would be like your vitamins. And the last one that we're going to talk about are our inhibitors. And this is probably one of the more important ones in our lesson, because this is actually going to affect the enzymes within HIV. This is going to be a way that they can be controlled, and people who have HIV can actually control the virus within their body. And that's when we can get into a discussion of antiretroviral therapy, or what we know as ART. And our inhibitors, we can have reversible inhibitors like competitive inhibition and non-competitive inhibition. We can also have irreversible inhibition and feedback inhibition. This topic of inhibitors is what's going to be shown to you in your next module.